Hi, my name is Emma Knapp, and today I'll be presenting biological sex-based differences in gut viral microbiome in people living with HIV. I would like to begin with my introduction. HIV is a retrovirus that attacks the body's immune system. It is primarily transmitted through sexual contact, blood transfusions, and shared needles, and currently has no cure and can progress to AIDS without adequate treatment. Now, HIV has infected 84 million people worldwide and has resulted in the deaths of 40 million people as of 2023. Additionally, HIV results in the massive depletion of CD4 T cells, resulting in immune dysfunction and fostering the progression and development of other infections. Now, the gut microbiome is composed of roughly 100 trillion microorganisms, primarily being bacteria, but also including fungi, viruses, and other microbes. Gut microbial composition and diversity is determined by many factors, such as genetics, diet, environment, and illness. Additionally, the gut viral microbiome, known as the virome, consists of gut viruses. As for gut microbiome connection to HIV infection causes significant imbalances to viral and bacterial communities in the gut, promoting systemic inflammation, gut dysbiosis, and impairing gut barrier integrity. Additionally, inflammatory GI states are linked to increased pro-inflammatory bacterial species, which promotes gut dysbiosis. Now, antiretroviral therapy, abbreviated as ART, is a treatment that suppresses the replication of HIV that boosts immune function while reducing risk of transmission. Approximately 15 to 30% of people living with HIV do not respond to treatment effectively, and many have difficulty restoring depleted levels of CD4 T cells. In some cases, ART has been found to induce patterns of gut dysbiosis in people living with HIV, which challenges the overall effectiveness of this treatment. As for the influence of biological sex on gut microbiome, globally the prevalence of HIV has been higher among women. Sex-based differences include HIV acquisition, viral load set point, immune activation, and how HIV persists in the body. Additionally, biological sex-based differences have been shown in both human and mice studies to influence gut microbial composition and diversity. As for the gap in research, historically marginalized groups have been excluded from study cohorts. Additionally, the National Institute of Health funded the NIH Revitalization Act of 1993 only 31 years ago, which finally mandated the inclusion of women and minorities in study cohorts. Due to this, there is a lack of studying women with HIV. Additionally, since my study cohort is from Uganda, it is important to note that women living in sub-Saharan African countries may not have accessibility to adequate therapies due to gender roles and their overall access to healthcare. As for the goals of this study and the hypothesis, the goals are to characterize the gut virome present in these samples from people living with HIV and controls, to examine gut viral correlation to biological sex in order to further understand viral diversity and abundance, and to contribute to research to develop biological sex-based therapies to improve the quality of life for people living with HIV. The hypothesis for this study is that viruses present in the gut will differ by biological sex in people living with HIV. As for the methodology, a subset of 65 samples underwent viral metagenomic sequencing, which included 28 male and 37 female Ugandan subjects. Of the 65 participants, 44 were people living with HIV, and of the 44, 21 were on ART. As for our ethical considerations, sample collection was approved and informed consent was received from all participants. As for the mentor versus student role, the mentor collected, extracted, and sequenced samples, provided the data for the study, and previously used the data to analyze bacteriome. Now, the student read 30-plus journal articles relating to the gut microbiome, performed statistical analyses independently on gut virome, created visualizations, interpreted results, and wrote the research paper. Here's my flowchart. First, matched unified stool samples were collected. Next, other information such as participant demographics, medication history, clinical symptoms, HIV RNA levels, and CD4 T cell counts were collected. Next, VLPs were prepared to enrich viral particles and reduce host bacterial contamination, and viral sequences were identified using Kraken-2. Next, the student used R statistical software for all statistical analyses and visualizations, which included the man-Whitney test for alpha diversity, Permanova for beta diversity, and heat maps were generated on normalized rates. As for our results, these graphs show beta diversity, which measures differences in gut viral community composition between samples. As we can see here, this is the PCOA plot for the all group, and this is the PCOA plot for the ART-treated group. These plots were generated using principal coordinate analysis based off Bray-Kerr similarity. Each point on these plots represents the respective gut virome of each participant, with blue representing male subjects and orange representing female subjects. Our ellipses on both graphs highlight our clustering patterns, and we can clearly see that men tend to cluster more tightly, while women are more dispersed across both PCOA plots. Additionally, Permanova tests yield significant p-values, which indicates that gut viral community composition differs by sex significantly in our study cohort. As we can see here, our p-values are 0.0043 and 0.0175, which are both less than 0.05, indicating significant statistical significance. Now these graphs show alpha diversity, which measures diversity of viral taxa within each sample. As we can see here, this is the all participants violin plots, while these are the ART-treated participant violin plots. Panel A for both shows observed number of taxa at the species level, while Panel B shows Shannon diversity, measured in the number of viral species as well as evenness. These are violin plots that show the distribution of viral species that have box plots showing interquartile ranges as well as medians. Man-Whitney tests were conducted and yielded insignificant p-values, meaning that gut viral alpha diversity does not differ by biological sex in both the all participants group and the ART. As we can see here, our p-values are greater than 0.05. Now these plots are hierarchically clustered heat maps that show relative abundances across all and ART samples. This is the all with 65 participants, and this is the ART with 21 participants. Columns indicate individual samples, while rows show our viral taxa at the species level. As we can see here, there's a color intensity bar which shows relative abundance, with red indicating high relative abundance and purple indicating low relative abundance. Additionally, dendrograms to the left cluster hierarchically by viral taxa as well as samples based off similarity of viral composition. Additionally, we have these colored bars at the top, which display biological sex, with men again being blue and women being orange. This allows us to visually assess whether samples cluster, and we can see here that there's minimal visual clusters associated with biological sex, which does not support our hypothesis. As for our conclusions, this study helps us draw new understandings of gut virome of people living with HIV in Uganda, an understudied region with the highest HIV prevalence. Additionally, this study can help us in the future be able to identify viral targets for biological sex-based therapies. It also provides novel research to the understudied field of gut virome. It also highlights the need for equitable sex representation due to biological hormonal differences that directly modulate the gut microbiome. As for future research, we would like to separate phage from eukaryotic viruses in order to independently analyze populations. We would also like to have a larger cohort with more participants on ART. Additionally, we would like to look at sex-based differences in a westernized cohort and compare it to our Ugandan cohort in order to see the role that population and diet plays. As for two of our major limitations, we excluded RNA viruses for this study, and we only had 122 participants with a subset of 65 for sequencing. Here are my references, and thank you for listening to my presentation.